Right, okay, so this is the fourth in my Flip Learning series on radioactivity. This one is focusing on the dangers and uses of radiation. So we need to know both industrial and medicinal uses of radiation. We need to know the difference between contamination and irradiation, and we need to understand the risk to living cells and also how to get rid of radioactive waste. So this slightly overlaps with some of the stuff in video 3, but I'm going to go through it again. The danger of ionising radiation on living tissue is because once the atoms in that living tissue become ionised, they react a little bit differently. This can cause copying errors in the DNA, which is known as mutations. So hopefully from biology you know that in order for cells to replicate, the DNA needs to copy completely. The ionising radiation can cause simple copying errors, like you've got here that T has become a C. That can cause abnormal cell growth, which can lead to cancer, but it's really important that you understand it's not always the case. So I've said it can cause mutations, which can cause abnormal cell growth, which can lead to cancer. We never say it does cause cancer, it just can cause cancer. But the higher your dose of radiation and your exposure to the radiation, the higher the likelihood. The other thing that's an important factor is your age. When you are young, your cells grow and divide very, very quickly. As you are older, the rate of cell growth and cell division reduces. So this is more dangerous to young people. So the exact problems of radiation in the body depends on which part of the body we're talking about. So in your eyes it can cause cataracts, in your thyroid it can cause cancer, in your lungs it can cause damage to your DNA, and it also causes quite bad burns and things like leukemia and blood diseases. So pretty horrible stuff. In terms of the dangers, we need to be sure of the difference between contamination and irradiation. If you think you know, you may wish to pause the video and write down your initial ideas before I go through the actual answers. So, irradiation is the process of exposing an object to nuclear radiation. So, if an object is irradiated, it just means it is in the presence of something that is radioactive. It doesn't mean that the object itself becomes radioactive. So, for example, if you have an X-ray, X-rays are ionising. If you have an X-ray, the moment you have the X-ray, you've got a slightly higher likelihood of a DNA mutation, but afterwards there's no long-term effects. A certain proportion of all carbon is radioactive, so if you stand next to anything living, you've got a small amount of irradiation, but there's no long-term effects here. We've got a lot of protection from cosmic rays. When I did the background radiation video in video 3, we said that cosmic rays make up about 1/8 of the natural of the radiation that is in the background. If you take a long haul flight, there is less atmosphere. If we're down here on Earth normally, there is less atmosphere on top of you if you're in an aeroplane, so you're temporarily exposed to a higher dose of radiation if you take a flight, which is why for a long time it was advised that pregnant women did not fly. So those are examples of irradiation. Irradiation being exposed to something that is radioactive. Contamination is a bit different. Contamination is the presence of unwanted radioactive nuclei. So the hazard there is from nuclei that are emitting alpha, beta and gamma. The level of hazard, so how dangerous it is, depends on the type of radiation, so whether it's alpha, beta or gamma, and also something called the half-life, which I will come on to in video 5. So the type of radiation and also the half-life of the radiation. So examples of this would be This is a Russian spy who was poisoned because his food got contaminated deliberately. He was deliberately killed by somebody putting some alpha in his food. Another example of contamination is after the tsunami, there was quite a lot of damage to the power stations in Japan at the Fukushima plant, and this allowed alpha, beta and gamma emitting nuclei to leak into the sea, and it's been traced all the way over to the States. Okay, so the presence of unwanted radioactive nuclei is contamination. So the level of hazard, as I've said already, depends on what type of radiation it is, the exact dose, so the exposure time, and also the half-life. There's a link here to practice thinking about which precautions you might need. Again, sorry, I didn't put a hyperlink, but you can pause that and type it in if you want. So if I gave you the choice of eating strawberries that have been irradiated with gamma, contaminated with radioactive rain after a nuclear disaster, or put straight in the box, which would you pick? A, B or C? I'm going to let you make your choice, then I'll go through the answer. Lots of people pick no. Lots of people pick C. As soon as they see the word irradiated and contaminated, they think, no, I don't want that, and they go straight for option C. But actually, the answer I would pick is option A. Why would I want option A? The reason for that is when you irradiate soft fruit, such as strawberries and raspberries as well, what it does is it will kill the bacteria and the bugs that is on the fruit, and it's got a longer shelf life. It doesn't do any permanent damage to the fruit, and it doesn't cause any long-term damage that could affect you, so I would probably pick A. So in terms of the radioactive waste, one of the biggest problems with radioactive waste is that it's got a long half-life. Again, we'll come on to that in the next video, but what that means is it's radioactive for literally thousands of years. There are a couple of different ways of disposing of radioactive waste. Sadly, sometimes it is encased in concrete and dropped in the sea. But we've also got in the north of England, I think it's somewhere called Sellafield up in the north of England. Basically, we've built a massive underground concrete bunker where it gets stored, but the exact danger depends on whether it's giving out alpha, beta and gamma, and how long it's going to be radioactive for. But like I said, that can be thousands of years, so we've got to think very, very long term, much longer than the lifetime of a human when it comes to getting rid of radioactive waste, and that's why some people are not big fans of nuclear power. So next we're going to look at common uses of radiation. So we've talked about all the bad bits, but when is radiation useful? So I've got six examples that commonly come up in exams. I'm going to run through the first three for you, and then I'm going to ask you guys to think about the last three. If you are watching this as a revision lesson, not learning it from scratch, you may wish to pause the video at the start of each one and write down what you can remember, what you think, and then watch it back. Okay, so this picture, what I'm going to try and bring out in terms of each example is which type of radiation would we use and why. So this picture is trying to represent the fact that we have got medical uses for radioactive isotopes. Okay, so what I'm depicting in this picture is a medical use of radioactive isotopes, sometimes shortened to radioisotopes. So what we use these for is to observe the function of different organs in the brain. In the body. What we actually have to do is pick an element that naturally collects in that organ. So for example with the lungs, that usually functions with a lot of oxygen and nitrogen, so we would pick those radioactive isotopes. We have to get the radioactive isotope into the body. We do that by injecting sometimes, ingesting, so getting somebody to eat it, or inhaling a gas. And then what we are able to do is watch how much radiation is given out from that organ, and we can observe the function of that organ. So these are called radioactive tracers or medical tracers. In terms of what source we would use, the important thing is the detector we're going to be using is outside the body. So the type of radiation we use needs to be able to get out through the skin, otherwise we're not going to be able to detect it. We also don't want it to be very ionising, because that could cause a lot of the problems I outlined earlier in terms of mutations. So with this technology, typically we are using gamma emitters, but occasionally beta, and never alpha, because it can't penetrate out of the body, and also because it's highly ionising, so causes, what have I done there? Sorry, I've just pressed one button. I need to press keep and then just put that back on the screen for you. It's highly ionising, so it can cause mutations, as we mentioned earlier. Okay, so that is radioactive tracers. Next one is if biological cells absorb radiation at a very, very high level, it can actually kill the cells completely. Most of the time that would be a negative thing, but sometimes we do want to kill cells if they are unwanted. For example, if we had a brain tumour. You can't operate on a brain tumour easily because it's inside the skull, and cutting open the skull would be very challenging and cause a lot of disruption to the person who is undergoing that surgery. So even though this isn't a knife, this technology is called a gamma knife. Why does it have to be gamma? We need it to be gamma so it can penetrate or get through the skull, and B, because we don't want to ionise the brain tissue in an unwanted way. What we have to be careful of is we've got the tissue that we want to kill, and then we've got the surrounding healthy tissue. So we've got the healthy tissue that we don't want to damage, and then we've got the cancerous tissue that we do want to damage. So instead of just taking one beam and pointing it at the tumour from one direction, which would give this healthy tissue a high exposure, we fire the beam in one direction and then another and then another and then another to minimise the exposure to the healthy cells and maximise the exposure to the cancer. So because each of these beams only crosses the healthy cells here once, all of that healthy tissue gets a low exposure, whereas where they overlap here at the tumour gets a high exposure. So that is called a gamma knife. And this is a photo of the like little helmet. Each one of these is a gamma source, each one of these, to get the effect of going from all of those different angles to minimise the risk to the healthy cells. The next one is a pacemaker. Now, a pacemaker is a device which is attached to your heart, and it's used for people who've got an erratic heartbeat. And what it does is it causes their heart to beat with a regular rhythm. Okay, so it regulates your heartbeat. Again, it's quite invasive to get this put in, so what you want to do is leave it in there for quite a long time. You can't be changing the batteries on this every 5 minutes. So in this case, the radiation is used as a source of energy. Okay, so the radiation is used as an energy source, because if you've got your unstable nucleus and then the radiation leaves, it's got kinetic energy, and if that kinetic energy is turned into electrical energy, it can be used to power the pacemaker. So, what type of radiation is used in the pacemaker? What would it be that's emitted from the unstable nucleus with kinetic energy? Well, it's actually alpha. The reason it's alpha is A, because alpha has got a mass, and we know that kinetic energy is half mv squared, so it's important that it's got quite a large mass. You may say, well, how could you use alpha in the body, because we know that alpha is very, very ionizing. The whole device is in a metal case, and alpha cannot penetrate metal. So because it's alpha, it's absolutely fine that it's inside the body, because it's inside the case, so it cannot penetrate the casing of the pacemaker. So this is a real picture of a pacemaker, and you can see that it's got the radioactivity sign on there. So I've given you a few examples of talking you through what the choice of radiation is and why. This is quite a common exam one, which is a smoke detector. I'm going to explain a little bit how it works. So you've got the radioactive source here, you have the source, okay, that is giving out radiation, and then you have a chamber of air, so source, and then the chamber of air under normal circumstances, and then over here the detector. The idea here is that when this chamber fills with smoke, so if that gets filled with smoke, less of the radiation is going to get to the detector, and then when this level drops, it sends a feedback loop to sound the alarm. So just thinking about how that works, which type of radiation do you think that would be, and why? So the answer is that it is alpha. We know that alpha has got a range in air of 3 to 4 centimeters, and so if it's sensitive enough that if that chamber fills with smoke and smoke particles are heavier than air particles, that will actually cause the drop in radiation levels that we would need to detect. If you use beta or gamma, the radiation level would not drop. So this is a picture of that. You've got your alpha source trying to pass through basically a stream of electrons. If smoke gets in the way and alpha can't get through, it breaks the circuit and the alarm would sound. So this is another really, really common example, which is using a radioactive source to control the thickness of a sheet material, like sheets of paper or sheets of foil. So you have the radioactive source on one side of the sheet and the Geiger-Müller tube on the other side, and again it is calibrated. Okay, so you're calibrated to know what the kind of typical level is. the way that this works is there's a feedback mechanism. So the machine knows what the calibrated value should be. If the value that gets to the Geiger-Müller tube suddenly drops and goes down, what does that mean about the thickness that's going through the rollers? Well, if it's dropped, what that probably means is the paper's got too thick. So what do the rollers need to do in response to that? If the paper is too thick, the rollers need to push closer together. So the rollers move closer together. If the opposite happens, if the Geiger-Müller tube value rises, what does that mean? It means the paper is too thin, so the rollers will do the opposite. The rollers spread apart. So which type of radiation would we use for this, and why? And the answer is beta. We know that beta is stopped by a few millimeters of aluminium. Alpha would, regardless of the thickness of the paper, alpha would all get stopped. So you couldn't use this very sensitive feedback mechanism that I've described at the bottom. Gamma is not stopped by paper or aluminium at all. So that would have the opposite problem of not really reacting to any of the changes in the thickness at all. So no gamma would get stopped. So because of how sensitive that feedback mechanism is, you have to use beta. Final one then, I think. The final one is using a Geiger-Müller tube to work out when a pipe has got a leak. So the way that this works is you deliberately infect or allow a radioactive isotope into the water supply. Okay, so if this is my supply here, at a certain point in time I allow radioactive isotopes into there. Wherever the leak is, here, there is the water is spread out of the pipework and into the ground, into the soil around the pipe. So if you know that there is a leak somewhere, what you do is you walk across the surface with your Geiger-Müller tube, walking along, and all of a sudden, when you get to this point, there's going to be a very high reading because there's of the water leaking out that's got the radioactive isotope in there, and there you would know, okay, that's where I need to dig my hole to fix the problem. So, which type of radiation and why? Well, if you use alpha, the radiation won't get out to the surface. If you use beta, it probably still won't get out to the surface. So in this one, it has to be gamma. Okay, so we're going to use gamma so it penetrates through the ground and reaches the detector. Okie dokes, so that is, I think, the last little one. So that is my little sequence on dangers and uses of radiation. I'm just going to show you the learning intentions for this section of work so you can be sure that we've done it. So, did we talk about industrial and medical uses of radiation? We absolutely did. We've discussed the difference between contamination and radiation. We've talked about mutations and the damage to cells and tissues, and we also talked about the difficulties with disposing of radioactive waste. So I hope that was helpful, and more from me soon.